I'm going to be talking about the DNA and how do we make the protein, all right? And let's go to it. So one of the things that is important is that the DNA or RNA, they're both genetic material, all right? So let's go ahead and take a look at the genetic material, their DNA first.
Sorry, is this chapter 3?
This is the end of chapter 2.
Okay.
All right. This is the end of chapter 2. You're going to get the rest later. So just bear with me. All right. But I'm going to probably cover some stuff that uh, may be part of a chapter 3, some stuff part of a chapter 2. I'm going to combine the two together. Okay. All right. So nucleic acid is DNA and RNA. In terms of the DNA, there are double helical structure. All right, DNA is a double helical structure, and it contains nitrogen containing base, a pentose sugar, and a phosphate group. All right, and pentose and the sugar group, they are known as a backbone of the DNA because they kind of like alternate and there's not a whole lot different from one another. All right, it's identical units. However, the nitrogen containing base comes in four different shape in DNA. So in DNA, you have adenine, which we abbreviate as A. You have guanine, which we abbreviate as G. You have cytosine, we abbreviate as C. And also we have thiamine, we abbreviate it as T. All right. Now, let's go ahead, before we look at the RNA, let's go ahead and take a look at the DNA and see it for ourselves. What does it mean by this? All right. So again, there are double-stranded DNA, and they are uh, providing instruction for protein synthesis. So here is adenine, here is the sugar group, here is the phosphate group. All right. Now, and here is a thiamine, sugar group, and phosphate group. So each of these are known as a nucleotide. All right. Now, if we wanted to take a look at the double helical structure, this one representing sugar, this one representing phosphate. Now you could see why is it called phosphate and the sugar are the backbone. You see this one phosphate, sugar, sugar, phosphate, you know, phosphate, sugar, and so on. So they kind of like alternate and they are combined. They are exactly the same, and that's called the backbone or the strand itself. However, there are bases in here that contains nitrogen. So earlier, don't worry too much about their structure, but you could see they contain a lot of nitrogen. That's why it's called nitrogen base. These are the base. So you have A, adenine, T, thiamine, G, guanine, and C, cytosine. All right, now we could go ahead and take a look at this one, and you'll see something that adenine and thiamine, they're always pairing up with one another. Adenine, thiamine, thiamine, adenine, thiamine, adenine. And guanine and cytosine, they pairing up with one another. So if you look at C and G, C and G, G and C. So they're always pairing up. So basically the number of G's is equivalent to number of C's, and number of A's are equivalent to the number of T's. No matter which organism you look at, from a bacteria to the human, they all have this universal numbers and similar DNA, okay, in terms of the letter coding, they're all the same. Now, the repeating units might be different. Like, for example, in here, we have A, C, 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 A. Maybe in another organism might be A, C, C, T, all right, or whatever. So there are differences, obviously, but the language is the same. It's made out of four alphabet, and how they come in together is basically making up really complex sentence that we need to figure out. Okay? So, what that means is if we know one strand, we should know the other strand. So, grab a sheet of paper, see if you can tell what would be the another strand. So, for simplicity, I'm just going to use this strand as phosphate and sugar backbone. I don't have to draw anything, all right. And I'm going to put in the nitrogenous bases right over here, all right. What I want you guys to do is draw another one. And based on the rule that I told you, T pairs with A, all right, and G pairs with C, all you have to do is figure out the other strand. Now, between them, you formulate hydrogen bonds. So A pairs up with T. So I got to put T in here. Next one, I got to put C in here. Next one, G. Next one, G. Next one, T.
Sí.
Next one, A. All right, good job. So make sure to do a couple of practices on your own. Put one strand, randomly assign these four letters, and see if you can figure out the other one. And it's really easy. All right, in between, you have hydrogen bonds in here. We already know what hydrogen bonds is. We looked at it when the hydrogen bonds was present in the connecting molecules of water with one another. Again, hydrogen is involved, and you have a slight negative and slight positive. They attract one another, and that's why you connect these bonds. These are hydrogen bonds. All right. Now, how does the DNA replicate? When we actually have a cell, let's say this is our cell, all right, and we have certain number of the chromosomes in here. Obviously, we have 46 chromosomes, all right, all right. So when the cell divides, mitosis, you learn in anatomy again, all right. So what happens is the end result is the same, 46 and 46 chromosomes. All right, so we look at one of these chromosomes. Here is one chromosome that is supposed to be, let's say, within a cell, right? This chromosomes is made out of DNA strands in here. And if we unwind the DNA strand in here, right, is actually double helical structure. And if it's double helical structure, then we can unwind that and bring it here, and you see how they are holding up with one another. However, in order to make 46 in here and 46 in here, I have to double this chromosome, right? In other words, I have to make the same amount of chromosomes as I have. In other words, I gotta have 92 chromosomes. So when they split, you get 46 each, and they are identical to one another. That's the ideal. This was called mitosis, and these are identical with what you start off with. So how is that replication occurs? How do we, in order to know how the chromosomes replicate, well, if we can figure out how the DNA replicates, we got it, because that's what happens. Chromosomes are made out of DNA. So for simplicity, I would like to erase these now and show you how we can achieve that. In the cell, the strands, they actually get separated from one another, all right? And one strand is gonna be used as a template. So all I'm doing, I'm just bringing this over here, and I'm assigning the same letter so you could see it a little bit better. And I'm gonna bring in this strand right over here, and I'm assigning the same letter again. So one strand is being used as a template, all right? And the new strand is going to be formed. This is also known as semi-conservative model, all right? So the new strand, I'm going to draw it the different color so you see it better. A pairs with T pairs with A, G, G, C, and T. Same thing with here. You have T, you have A, you have C, you have C, you have G, you have A. Again, there are hydrogen bond in between two, and basically now we can take a look at it. Tell me if you see any difference between here and here. Look at the letters. It's identical to one another. Are they identical here? It is. Look at it. TACCG, ATGGCT, and exactly the same thing. So this is the original cell right here, part of it, within a particular chromosomes. And this happened to be in another cell. This happened to be in another cell. So what happens is later on, these come apart, and again, they go into a different cell. That's how you duplicate the chromosomes. The point is that the DNA is identical when you compare your DNA of your skin cell to the DNA of your, let's say, cheek cells. They're identical to one another. This is actually how criminals get caught because, let's say, a person is a suspect, left traces of a follicle of the hair on the crime scene. They analyze the hair. In the hair, there is a root in here. Here's the hair, there is a root in here. That consists of DNA. All right. And once you add appropriate content of several different chemicals and so on, you could separate the DNA and you get and feed it into a complicated computer, and it will give you the results of their DNA. What are the letters, for example, on a specific area of the DNA? And if that matches from the hair from the suspect's DNA, that means they are identical, and that's how they get caught. And this can be used in a court of law and to convict a person and so on as well. Uh, a lot of people unfortunately got caught wrongly. They thought they were in, they committed the crime. They went to the prison. Back in the day, they weren't that much DNA technology available. So they served several years, like, 10, 20 years in prison. And then now that the DNA evidences are more advanced and so on, it was found that, hey, the DNA of the person who committed a crime does not match this person. And now they are freeing them. Can you imagine wrongfully you end up in the prison? It's it's crazy. And as you know, if you're a certain race, you might be actually more uh, you know susceptible towards that. It's all unfortunate problems, but anyways, that's the point of doing DNA and forensic science. But I just wanted to tell you that how they are replicating, so you have a better idea. Now, if we analyze, let's say, a small sequence of the DNA, and you find out that you had 30% thiamine in unknown DNA. Let's say this is my DNA. I only analyze a little bit of it, and that contained 30% thiamine. Can you find out the percentage of the cytosine? Well, let's take a look. Remember, the number of T was equals to A. The number of G is equals to C. So if you have 30% thiamine, that means I have to have 30% adenine. So right now, I'm added 30% plus 30%, that equivalent to 60% is gone. 60 out of what? 100%. Per 100. So that means 40% is remaining. Now, because cytosine pairs up with guanine, yes, Guanine gets to get 20% and cytosine get 20% because 20% is half of this 40%. So if you add all these 20 plus 20 plus 30 plus 30, that gives us 100. All right. So yes, it's possible. Even if you have the percentage of only one of them, you should be able to find the percentage of all of them. All right. Yes, it is possible. All right. So moving right along, what about the RNA? RNA is only one single stranded. All right. And the rule is a little bit different now. In RNA, you have uracil. We do not have thymine. So RNA, it has uracil, guanine, cytosine, and you do not have thymine, but you have adenine. All right. Instead of thymine, there is a uracil. DNA does not have a uracil. All right. So if that is the case, then we can go ahead and take a look about the base pairing rule and figure out how we can figure out the DNA. Now, central dogma states that DNA can give us a messenger RNA. Messenger RNA can give us protein. I'll go over that. But right now we are at this level because we looked at the adenine, thymine, guanine, cytosine, how is it replicates, there's double stranded and so on. Now, let's say I'm only drawing a single strand because let's say this is called a coding strand. One of these is called the coding strand. This is called coding. Remember that all the information to make the protein comes from the DNA. In fact, DNA is very important because all their instructions is related to that. You are tall because you have certain genes, all right? You have a tall gene, for example. Your hair might be curly because you have the gene for curly hair, right? So these instructions are embedded in the DNA level, all right? But we don't use both of these strands. One of these strands is a coding strand, and that is the one that is being used in order to make the protein. Well, how do you know which one it is? Well, the cell does know, and there's a lot of other proteins and complex enzymes and the letters that it might be too advanced for me to go over, and also it might take a little bit time. However, let's say coding strand for this, I just might want to make it simple. Another strand, you already know, I'm not going to draw it up here, but that's not going to be used. Usually, whenever there is a TAC and so on, usually that's the one that is a coding strand. But there is a lot of proteins and enzymes and transcription factors and things like that comes into a play. But we want to make it as simple as possible and give you guys some activity to do. All right, now in messenger RNA, remember, at the DNA level, if you have A, right, there is no thymine, so at the RNA level, that would be U. If you have C, that would be G. If you have T, that would be A. If you have G, that would be C. So having said that, we can easily figure out the messenger RNA, which is actually one strand. So let's get to it. So I will explain further a little bit later on, but it looks like this is A, U, and G. And the next one, C, C, C. And the next three, it looks like is G, G, G. And the next one looks like it's G, U, A. Let me just modify it a little bit so that way. Let's add three more. And the RNA level, you know what it will be, BU and UGA. Okay, all right. So this is called a messenger RNA, and basically what happens is that there are enzymes involved that kind of like zip through this and add in this nucleotides one by one as they go through and zip it. And it would be a good idea to look up on the YouTube transcription. All right. Maybe we can do that later, all right? But this is called a transcription. Transcription is making the RNA to the DNA. So any DNA that I will give you, it doesn't have to be this one, but any DNA that I give you, I expect you guys to know how to transcribe it, all right? I'll give you activity towards the end. Maybe you can do it. And to just get a little bit of practice. All right. But right now, what I like to do is go over a few more things and see what happens.
I'm
All
sorry,
right.
Professor. I have a question. I'm so sorry. Did you say transcription is making DNA to the RNA or the vice versa?
No, making a messenger RNA from the DNA is called transcription.
Okay.
All right.
Thank you.
You're welcome. All right. So now what we wanted to do is find out how we can do the translation. Now, transcription happens inside of the nucleus. So this part right over here, right, it's inside of the cell nucleus. But this whole thing right over here is inside of the cell. So there is another process, it's called translation. And translation happens where? In the cytoplasm. This is called cytoplasm. All right. So the next step is what we wanted to do is figure out how do we do the translation, which is actually very, very simple. If you guys pay attention, we can do this. It's not that difficult, to be honest with you. All right. But you need certain things. All right. You need certain things. So what we're going to be doing is trying to look for how we're going to do the translation. All right, so before we go on to the process of translation, all I need to do is figure out what amino acids, proteins are made out of amino acids, all right? And it turns out that each of these three letters, they code for one amino acid. These three code for another amino acid. These three code for another amino acid. These three codes for another, etc. So the protein is combination of these amino acids. So all I have to do figure out is what are amino acids in here. So for that, I need to look it up. All right. You could Google this at any time, but for the sake of doing things now, why don't we take a look at this one and go back to see if you can figure out. Now, the first one, it was a AUG. So these three letters are abbreviation for a type of amino acid. This, for example, met is for methionine. all right? So you could put meth underneath that and go from there. Let's see where we were. All right, so this AUG codes for meth. Next one is CCC. So again, I gotta go, where do I get this? You could look at up in the Google, you could just put in universal code image and it comes in. So it looks like CCC right here. It's code for proline. But then again, how do I get this very fast? All right, well, let's take a look. Where was the, all right, CCC. So this is the first base. This is the second base. This is the third base. And I could put proline in here. All right. Now let's do this together. My first base is here, second base here, third base here, because they are being read 3 at a time. So it looks like they're all the same, G, 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 G. So if I go back to the universal picture, all right, first letter was G. So notice that all of these starts with G. All of these that are in this particular row starts with G. Second letter or second base was a G as well. So notice U, C, A, G. All these column starts with U. All these column starts with C. All right. Look at the second letter. You have to look at the second letter, not the start, but the second letter. Second letter A, second letter G. So far G, G. And the third one is also G. So where all the line intersects, I get triple G right here. All right. So therefore, I know what I have. I have glycine. So if you want, you could pause the video or see if you could find the next one is GUA. All right, GUA. We need to find GUA. Can you find GUA? Yes, it is valine. All right, or val. So we can go ahead and put that one in, and we can put val. GUA was valine. Now the next one is UGA. UGA happened to be a stop codon. That means it doesn't code for anything. Let's see. See, UGA, it says a stop. What does that mean? I mean, that's the time that I need to stop. It doesn't code for any amino acid whatsoever. So what happens then? Well, what happens then? You already make your amino acid, they might fold or have certain function or whatnot. We started at the DNA level, we went to the RNA, and then we made the protein. Protein is chain of amino acid. This is chain of amino acids. Now, if you have corrected letters, corrected genes, your protein is going to be corrected. If you have faulty gene, maybe the protein will be faulty, like sickle cell anemia. In sickle cell anemia, there is a faulty gene, all right? That's why the hemoglobin in the red blood cells are not the same shape. So as a result of that, instead of having a nice round cell, you have sickle cell, all right? So this is called translation. Translation, it means like, you know, it's like a new language that needs to be translated to the protein language, all right? So that's how you do it. So as a practice, why don't we do another one? This time you are on your own. I'm just going to show you the coding strand at the DNA level. All right, you have TAC, GGT, AGC, GAT, and let's make it simple again. In reality, it's a lot longer, but let's say here you have a DNA coding strand. How do I know if I have DNA? Well, thiamine only exists in DNA, so I gotta have that. Now I got to figure out the messenger RNA. All right. So you need to write that down. What is the messenger RNA? Once you write that down, then you need to use the coding, universal coding system, and figure out the DNA, the protein. But really quickly, go ahead and see if you can figure out my messenger RNA. In other words, transcribe this. After that, you need to tell me what is the protein, what are the chain of amino acids, and go from there. All right. Now, I'm going to give you guys a little bit time to read this, write it down, so that way I can reveal the genetic code. I hope everybody had enough time to write it. Here's the genetic code. Use the genetic code now and figure out the protein. See if you can do it. All right. And then again, on the chat, if you want to, you could type in the answer that you got. And then if you got the similar answers, then you could figure it out. If you guys chat among one another while I give you a lecture, you will be confused, okay? And I do not want any negativity, folks. I don't want you guys to be complaining or any negativity. Oh my God, I can't understand anything. What you're doing, not only you're yourself panicking, but you're also making others panic. All right. A person that, let me pause the video. Okay. So there is a molecule known as adenosine triphosphate, and this is a usable energy for the cell. Cell requires a lot of energy to do the things that it does. All right. So for that, you need ATP. ATP comes in from the mitochondria. In anatomy, you learn about this organelle, and you probably learned that was a powerhouse of the cell. But in particular, it generates ATP. In terms of the ATP, I wanted you guys to picture that adenosine triphosphate is on top of the hill. All right. This is like your bicycle on top of the hill. All right. It takes time and energy to go up, but as you come back down, you can break these bonds. These bonds are high energy. So what that happens is you're going to get adenosine diphosphate and plus another phosphate. What we did? What we did was we break the bond. So energy is released. This energy can possibly used for the cell to do something, to, make proteins, to, process something, right? Or in this case, letting you coming down from the top of the hill. There is another bond right over here. Again, you can get adenosine monophosphate now, plus two phosphate groups, two bonds are broken, energy releases, right? But at times, in order to get up there, we talked about the potential energy. So it requires energy to put in here to get to the adenosine diphosphate. And then another phosphate group, it requires energy to get up there. So what do we use this ATP for? As I mentioned earlier, we can use it. This is the molecule looks like. This is the high energy bonds that I just told you about. All right. Don't worry about the particular structure. That's not really relevant at this point in time. But you could process something. You could contract the muscles, for example. You could add things together in a chemical reaction. Sometimes you need energy. You could perhaps transport stuff outside of the cell or bring in material inside of the cell. So those are ATP at work. They are working, they are doing something. All right. Now, going back to the process of transcription and translation. All right. Uh, Let me see what time we have. Let me pause at this point in time. What we are looking at is chapter 3, you guys. And the chapter 3 consists of the cell and its organelles and what do they do. And also it consists of more important stuff in regards with transportation, what material and how material gets in and out of the cell. And basically that's where it's building up, okay? So um, first, I would like to go over a few things with you, and then we'll get to the transport mechanism. All right. So first thing is first, I would like you guys to understand that there are, let's see, plasma membrane. So here is a generalized cell. Cell theory states that all cell must come from pre-existing cell. There are a lot of department and organelles within the cell, but right now we're looking at the membrane. Right here is the membrane, plasma membrane, which is phospholipid bilayer. We mentioned that already, all right? But the plasma membrane, it separates what's inside from what's outside, all right? Outside of material, we call it extracellular material. Extra means outside, exterior, things in that nature, all right? Each cell is different, so by looking at this cell, it does not mean that our cells is only this way. No, muscle cell completely look different. Your nerve cell is different. Your sperm cell is different. Your red blood cells are different. So not necessarily, but again, the cell membrane plays a role in cellular activity. What material can come down, what material can go out of the cell. And there is a lot of glycoprotein or glycocalyx that is on the surface as a biological market that cells can recognize, especially the immune cells. Uh, there is a bilayer, as we mentioned, and if we go ahead and take a look at, you could see a smaller area of the membrane have been expanded over here. So you could see a lot of glycoprotein, things that are useful for biological marker. You could see double layer. Here's one layer, here's another layer. And again, we discussed the in between, you have the lipids portion of it, and non-polar tail, which we call it the hydrophobic tail, because they look like tail, and this circle looks like head, but this one has a phosphate group, so that's what we discussed earlier, right? There's also cholesterol there for maintaining the fluidity of the membrane, but really what it is, there is a lot of other structures in there. Take a look at other structure. These are protein. Protein could be completely embedded inside. So for example, if you're inside of the cell or you're outside of the cell, you could see the same protein. So this one I can see here. If you're inside of the cell, I could see it too. So these are called integral protein. Peripheral protein, you may not be able to see it from both ends. So it might not be embedded completely. So that could be the case too. So for example, if I have the protein only this part right here, if I'm outside, yes, I could see that. But if I'm inside, I cannot see anything in this side. So these are called peripheral protein. Notice that they're pointing only to this portion, which is attached to another protein that it was integral protein. But anyway, this is only on one side. Regardless, these proteins are important. They do certain activities. Again, we wanted to take a look at 6 categories of these proteins and what they do. And because they are attached to the membrane, we call it membrane protein. let's go ahead and take a look at the job of the membrane protein. Number one is transport. So earlier, if you look at in this slide, you could see there is a little gap in between. This little gap, some substances that are small can exit through this gap and end up outside. Or vice versa, they could come in through this gap inside of the cell. All right. So what are they there for? They are there for transport mechanism. They can transport things, sometimes using the energy, sometimes without using the energy. So their job is for transportation of substances, either bringing inside of the cell or taking things outside of the cell. All right, they do have enzymatic activity. Enzymatic activity. All right, so they can act like an enzyme. You could see this is the substrate, now is a product becomes a substrate, again, they're making another product. But the difference is that these enzymes are attached to the, uh, membrane. The next one is they act as a receptor, like they detect things. We talked about, certain protein that they are chemoreceptors or whatnot. These are receptors on the surface of our cells. They could detect hormone, for example, or other things. All right. Next up, we have intercellular adhesion. So if you have one cell right here, you have another cell right here, intercellular adhesion, that means they glue things together. If you pull on your skin, on your wrist, for example, the skin does not come apart. Why? Because each cell, inter means between, cell, adhesion, like glue, all right? They act like a glue, they join in one another. They can act as a cell-to-cell recognition. Again, there are a lot of biological markers, like this one, for example, and another cell can come in and make sure that they are belong to you. If not, they can attack. And sometimes you probably notice or heard that during organ transplant, sometimes the body rejects a particular organ, and that's what happens. All right. Next up, we got attachment to the cytoskeletal. Cytoskeletal is kind of like a frame to a house. So they can attach to them, anchor them, so they provide a structural stability for the cell, so the cell doesn't just collapse. All right, so those were the 6 category of the membrane protein. All right. Next up, we can go ahead and take a look at okay, membrane junction. There are 3 different type of membrane junction. One is called the tight junction, which is again is kind of like a intercellular adhesion that we discussed. That's the same thing. All right. So my pen is not working. Okay. So you have desmosomes and you have gap junction. And all of these, again, they help out in order to attach, in order to support, in order to do different things. My pen, all of a sudden, it froze, you guys, so I cannot write things down. Let me go ahead and give you guys, again, let me work on this one. And let me pause the video again.